Hello, welcome to the week 9 extra live session. Sorry for postponing the class today. It was supposed to happen on last Friday, 6 to 8. I got an urgent medical emergency, that's why I postponed it to Tuesday. Yeah, I hope you remember and went through all the 5 week lectures classes. So we were in the last week, week 8, we were discussing about neural system and special senses. We especially talked about hearing, about ears, and about olfaction, how the olfaction is happening and how the brain is recognizing that sense. This week we are mainly going to look at the other sense, which is about our vision, how we are able to see, how our eyes are able to determine shapes and respond it to the based on the intensity of the light, and how our neural system is coordinated with all these things, and they are making the vision possible. Hi, Austin. So I was just giving the brief messages of what we were like what we have learned in the this week 9. Mainly, we have seen the about the eyes and eyes, how eyes are coordinating its activity with the neural system, and how they are performing, how our brain is able to analyze, respond based on the intensity, and analyze the images as images. Did you went through the lessons? Hi, Austin. Yeah. I will start the class again with my self introduction. I am Harsha. I am a PhD scholar. Fourth year of my PhD just started. I am doing my PhD in Department of Biological Science in ISER Barambur, under the guidance of Dr. Asalvi Bhargavikru. So that's about myself. I am also one of the TA to the course Animal Physiology taken by Professor Mainak Das. Yeah, so I'll share my screen, okay? Yeah. So welcome to the class again. So we were telling about what are the brief summary of the last lecture. So first lectures are mainly talking about anatomy of the eye, followed by eye lens and about cataract. And in the lecture 3, sir was talking about structure of retina, and followed by in lecture 4 how the image formation is happening and how the image processing is happening in eyes. Finally, sir talked about mechanism of photo processing by rods. So, if you remember, neural system, especially neural system after the muscles, it was the longest lecture. We started in each session. We went through I think first lecture we it was introduction, second lecture and also talked about integumentary system. Second week we talked about skeletal muscle. Then the third week again mechanism of. Then third week towards the end, I think we started with neural system. This is week 9. So by this lecture, we are ending with neural senses, and we are now going to look at the other organ system. Okay. Yeah. So what was the first lecture summary? It was regarding anatomy of eye. So eye looks like sir told us eye can be compared to virtually to a camera, right? Camera also have a aperture where light passes through. Aperture is the part where we can determine how much light we can allow inside or not. Aperture can be adjusted. So cornea of the eyes can be compared to aperture. Then inside, if we are looking, there is a lens. Camera also has a lens. Our same similar structure is also a lens. And then we have inside where the light falls on and the photoreceptor cells are present. That layer is called as retina. So the processor is known as CMOS sensor in camera. So retina can be virtually compared to a CMOS sensor. Okay, so that is the comparison between eye and camera. Then anatomy of eye, if you are looking, there is cornea, then there are lenses. Lens is very transparent color of transparent structure. In the next, the in the second week of ninth, in the second class of 9 weeks, are actually mainly talked about what are these lenses, how they are becoming transparent and all. And inside the so outer there is a sclera, white layer that we are seeing. That is sclera, then cornea, then lens. Then inside there is different. It's a there are two partition in the eyes. The sclera or cornea to lens part there is aqueous humor present, and in the lens to retina we can say it is present with vitreous humor. These two partition is inevitable for the maintenance of pressure in the eyes, eyeball, and to do all the functions. It's inevitable. Then if you are looking at the inside part, then there is retina. As I said, there is all the retinal pigment or epithelial cells, photoreceptor cells, everything is present on that layer. Okay. Then sir also told about us the eye donation. So I also used to think like sir, as he told in the class, when we agreeing for eye donation, maybe they are taking the entire eye. So no, that is not the case. When we talk about eye donation, it is just the donation of the cornea. So that's it. Then anatomy of lens. Lens were first formed with cells only. Like there are lot of small cells found inside that. These cells in the initial part, initial evolution in time, it has all the cell organelles, mitochondria, nucleus, ER, Golgi bodies, sarcoplasmic reticulum, everything was there. But then what happened? Sorry, here endoplasmic reticulum, everything was there. Then what happened? Like our RBCs in the blood, where also initial part all the organelles were there, but in the due to accommodate or design to its enable to do its function, most of the organelles will start shrinking and hemoglobin pigment will get filled up. Like that, here is also similar thing is happening. Eventually, organelles will go, and all these cells will get remove get filled with increase in level of 15 allox lipoxygenase. So what is lipoxy? Lipoxy means lipid digesting. So we know our cell membranes, even the cell membranes, all the organelles are also membranes are made of lipids. So they digest the lipid. That's how it digests all the organelles. And then there is another transparent protein fills up in that place. That protein is called as alpha crystalline, which we get to know in the second class. So this alpha crystalline will come. Okay. Uh, Now talking about second, it was I said RBC structure was also similar. The evolution of RBC was also similar to like the lens. Hemoglobin filled in RBC. Here alpha crystalline get filled in filled inside the lens. So structure of lens cell, if you're looking inside, it's fully of alpha crystalline. And what happens in cataract? We know cataract. Uh, the lens in the lens there is some due to the misfolding of this alpha crystalline protein, that area will become like blind spots. So the due to the misfolding of protein, that's why the people are unable to see clearly. So it's a age related disorder maybe mostly. So in mostly as age progresses, cataract is common. So for that people do lens replacement surgery and all. Okay. And in the third week, it was regarding structure of retina. So here it's an important part where we are how our eyes are able to see, because there are photoreceptor cells. Photo means light. So photoreceptor cells present, which are rods and cones. So sir drew the image. Rods looks like rods and cones look like cone shape, and how they are arranged in the retinal layer. We like this figure shown in here. Let me take the laser. Yeah. So this figure is seen in here. If you invert it like perpendicular, then it is arranged in the retinal layer like that. Um, so these rods and cones are the cells that takes up the light information or whatever they like that gives the information to our nerves, and nerves gives it to the photocenter of the brain. That's how we are able to see things. Okay, so that's about. Then also there are other than retina, there are other than some cells like horizontal cells, amacrine cells, bipolar cells, retinal ganglion cell. Each one is followed by next one, and are arranged inside this. Okay. Now, talking about image formation and processing. So, especially I told rods and cones are responsible, but unlike other neuronal electrically active cells, here both the rods and cones are electrically active, which means they can they have the capacity of generation of action potential and everything. But when we studied about everything else, we studied that the resting membrane potential is minus 70 millivolt. Unlike that, here the membrane potential, if you are looking at this layer, it will be minus 40 millivolt. So that's the interesting part. That's why Sir told about we always think, okay, when light comes to eyes, these receptors get activated. That's why we are able to see. But that is not the case. When light comes, if you are looking at the resting membrane potential, that is minus 40. And during the dark only, the resting membrane potential changes. So, the like whatever we think, like because of light, the action potential is generated. That is not correct, because in the light, resting membrane potential stays minus 40 millivolt only. So that's why Sir draw this figure, the black dots. He explained it very correctly. We can see like this also. So we can draw the same things analyzed by looking at the black dots also. So some concept like that. Then talked about rods and cones. So always remember, cones are responsible for the color vision, green, blue, red, whatever the like colored vision. It is concerts are responsible, and that varies in multiple organism. For example, cats and or have like a very great vision during the night, so that changes. Um, rods are the cells that respond to the like black and white vision. They doesn't have any colored vision activity, but they can process black and white vision, or they respond based on the intensity of the light. So we can say like that. Now, what are the chemical changes? Both these cones and rod contain two pigments. Rod contain rhodopsin. So the rhodopsin is made of opsin and retinal. And again, when we go to first introduction lecture, the vitamin A, that is a source of retinal. That's why our parents tells us to eat more carrot and all, because that is the good vitamin A source. So we get more retinal, the which can increase our vision. So our parents always tells to eat more vitamin A containing foods. So what are the chemical changes happening whenever light is like coming? So rhodopsin changes to opsin and retinal, and as a result, what happened? Then it gets activate phosphodiesterase enzyme. So activated phosphodiesterase enzyme will go, and then it will tell the cyclic GMP, which is the gate opener, to remain dissociated. So no more sodium channels will go inside, and thus the resting membrane potential will be maintained. Okay, so also there can be why the eyes have minus 40 resting membrane potential, because always the sodium channels and calcium like things are more. That's why it always maintains the resting membrane potential due to the positive ion stimulate. That current is known as dark current. So due to the dark current only, it is able to maintain a resting membrane potential of minus 40 millivolt. Yeah, here it is written. Okay. So remember, due to dark current, it's able to maintain. Yeah, now let's go to some questions. Okay. Sorry, which of the uh, which of the following is not a part of the eye? Option A, retina. Option B, cornea. Option C, iris. Option D, glomerulus. Not a part of the eye. This is very, I think, this is easy question only, I guess. Which of the following is not a part of the eye? Question you can type answer in the chat box also. If you know, if you don't know, so I will discuss which of the following is not a part of the eye. I think I accidentally opened the answers also, answer tab also earlier. Any idea? Yeah, very good. Uh, so question is saying B is not a part of the eye. Can you say glomerulus is a part of which cell in our body type? I'm really sorry for my sound. I got a cold. That's why my sound is like this. Uh, yeah, so the correct answer is glomerulus, and glomerulus is a part of nephron. Okay. One thing I forgot, I actually forgot to connect to my headphone. 1 minute. Yeah, so we were saying glomerulus is a അല്ല object both objective subjective ഉണ്ടായിരിക്കും. ലൈക്ക് ഞാൻ ഇംഗ്ലീഷിൽ പറഞ്ഞോട്ടെ കാരണം യൂട്യൂബിൽ ഇടുന്നത് കൊണ്ട്. So the technical details of exam, you can just send a mail to the NBTL who coordinate, like who sends you mail. You can ask them. Actually, we don't get any information regarding exam pattern and all, so you can ask them. Okay, I don't know about the technical details about the exam. So for how much mark it, I know it will be subjective and objective. I guess. Yeah, and how much part will be objective? Yeah. Now you can hear me, right? Yeah, I will again present the screen. I think my can you tell me something? My earphone is connected, but I don't know whether it is connected in the meet or not. Presentation. അത് ഞാൻ സ്റ്റോപ്പ് പ്രസന്റേഷൻ കൊടുത്തു. ഞാൻ ഇത് ടെസ്റ്റ് ചെയ്യുമായിരുന്നു. ഇയർഫോൺ. I was testing my earphone, that's why. Where are you from, Kerala? Where in Ernakulam? Oh, okay, okay. I know. My house is in Kottayam. Okay. Sorry, there is a rule. Since it will be uploaded in YouTube, I can only speak in English. That's why I'm speaking in English, and it's recording. So that's what. I will share my screen again. Yeah. So going to the next question. The area from which the optic nerve leaves the retina is called. Option A, sensory spot. Option B, blind spot. Option C, retinal neuron area. Option D, descending region. The area from which the optic nerve leaves the retina is called. So I'll wait for each, after reading each question, we'll wait for 60 seconds. And if you know the answer, you can type it in the chat box. Otherwise, I'll discuss anyway. Don't worry. Okay. So the area from which the optic nerve leaves the retina is called optic nerve starting. So when Sar drew a image of eye, he showed that there are lots of nerves coming and that everything leaves at that point. So particularly in that particular point, there is no photoreceptor cells present also, and that spot is known as blind spot. Okay, that's a no photoreceptor cells are present. That's why it's known as blind spot. So correct answer is option B. Okay. So in this area, there are no light sensitive cells. So this part of your retina you cannot see. Why? Because all the nerves will be leaving. So should I go back and see you that figure where sir drew? Anyway, it will be coming. So that time I'll show that image. Okay. Which part of the eye is majorly donated? This we discussed during the introduction part of the class also today. Which part of the eye is majorly donated? Option A, retina. Option B, cornea. Option C, iris. Option D, glomerulus. So which part of the eye is majorly donated? Good, Austin. Hmm. So now time is 6:23. I'll wait for 6:24. So the correct answer is cornea only. Very good. So cornea is the part that always donated in that donation is generally known as eye donation. Uh, but that doesn't mean that entire eye is taking out and donating. It's only that part of cornea is donating. Cataract is caused by the cloudiness of dash. Option A, aqueous humor. Option B, ciliary muscle. Option C, lens. Option D, vitreous humor. Cataract is caused by the cloudiness of dash. In cataract, which part is affected? Yeah. So it's 6:24. For the people who are going to see in the YouTube, that's why I'm saying 1 minute. So 6:25. Yeah, lens is the correct answer. Okay. So cataract is caused by the cloudiness of lens. Why it is caused? Due to the misfolding of alpha crystalline protein. Okay. And eventually that get folded up so that spot become non-transparent or yellow color like black color. So no light's able to passes through and vision gets problem, so no correct image processing will be happening. A person with 20 by 50 vision has dash. This we haven't discussed, but I think you have learned about these disorders, and you know some people have different types of lenses. Most of the people who has glasses, maybe some have convex, some have concave, some have cylindrical lens. So there are multiple kinds of disorders in the eye. So it's that kind of question. A person with 20 by 50 vision means what kind of disorder he or she has? Myopia, hypermetropia, option C, both A and B, option D, none of the above. Okay, question is saying myopia. So 20 by 30 means, uh, so it's a vision is worse than the average, right? So 20 by 50, why it's written like that, which means that the distance at a 20 feet away, we can read letters. Most people see that letters from maybe 30 feet. So, which means nearsightedness or myopia is when close objects look clear, but distant objects are blurry. So, we can see whatever the near things. For example, this bottle is here. So, I can see if I have a myopia, then I can, there is no problem. But then if I want to read some bus is coming, so I want to read the what is written, the place written on the bus. So in that case, if they don't have a lens, then it's difficult for them to read the things at the distance. So if it come closer only, then they will able to read that clearly. So myopia is things they will able to see things clearly which are nearer to them, but the far things they cannot see clearly. The focal length of the lens increases when ciliary muscles dash. Option A, contract or are strained. Option B, relax. Option C, focal length of the lens is fixed. Option D, focal length of the lens does not depend on ciliary muscle. It depends on the size of the retina. So what is the correct option? Is there is any connection between focal length of the lens and ciliary muscles activity? Is there any activity? Contract or are strained. Okay, let's see what is the correct answer. Correct answer is relax. Okay. So the question is increases, right? Focal length increases. Focal length increases means ciliary muscle must be in a relaxing position, then only focal length of the lens will increase. So when ciliary muscles contract, that means focal length is decreasing. Okay. The eye accommodates by assuming a lens shape that has a longer focal length. So far, distant objects, the ciliary muscles relax. So whenever we are seeing the objects that are far away from us, in that condition is the ciliary muscle relaxing. And that time lens returns to a flatter shape because ciliary muscle in the relaxed condition and lens take a flatter shape. Uh, so this decrease in curvature. So that means lens take a flatter shape means the decrease, the focal length of the lens is uh, this decrease in the curvature of the lens. So curvature of the lens is decreasing, which means to the curvature is decreasing, which means focal length is increasing. Okay, don't confuse by connecting with curvature of the lens and focal length. Curvature of lens becoming smaller means focal length is increasing. Okay, so focal length is increasing means ciliary muscle is relaxing. Retina consists of photoreceptor cells, rods, and cones. Rods detect dash and cones detect dash. Option A, brightness of light. Rods detect brightness of light and cones detect intensity of light. Option B, rods detect hue of light and cones detect color of light. Option C, rods detect hue of light and cones detect brightness of light. Option D, rods detect intensity of the light and cones detect color of the light. What is the correct answer? This also we discussed during the introduction. What is detected by what? Any idea? If you have any doubt in the explanation also, please stop and feel free to ask. Okay. For this question, do you have any idea? Okay. Hue of the light and brightness of light. Okay. So the correct answer, I said, when we with cones means cones are there are depending on the color we have like three types of cones. For cats maybe it's because it will be four. So cones major function is to detect the color, detect different types of colors. And the road is to, it doesn't have to do any color, it doesn't have multiple types and it cannot analyze color types, so it majorly see black and white only. But at the same, it respond we discuss, right? When light comes, rhodopsin will disintegrate into opsin and retinal, so all the mechanism happens. So that is based on the intensity of the light. So correctly, answer is intensity of the light and color of the light. Okay, rods detect intensity and cones detect color. In the dark, the photoreceptor is depolarized due to an influx of dash through open ion channels that are gated by dash. So it's again about dark. Remember, we talked about dark. It's not like always, even though light is in response to light, rhodopsin is responding. That time CGMB is getting closed only. But in the dark, photoreceptor is depolarized. Depolarized. So depolarized is an important part. Always remember, during dark, photoreceptor is depolarized due to an influx of dash through open ion channels that are gated by dash. Simple question is, who is the gate opener? Second question is that. The first question is, which ions are responsible for that? Which ions are coming when gate opens? Option A, sodium, calcium. Gate opener is AgMb. Option B, sodium, potassium. Gate opener is calcium. Option C, potassium and CgMb. Gate opener, calcium. Option D, potassium and calcium. Gate opener, sodium. What is the correct answer? Do you know, Austin? Sorry, did I change it? Sorry. You must have seen the answer, no? You can type that also. Yeah, correct answer is sodium and calcium. Depolarizing is happening due to the influx of them, and gate opener is CGMB. Very good. Okay, that's why the dark current maintains that resting membrane potential in photoreceptor cells at a minus 40 millivolt. Remember that also. Okay. So in the dark, photoreceptor is depolarized due to an influx of sodium and calcium through open ion channels that are gated by CGMB. Photoreceptor has high levels of CGMB when it is in the dark. For refractive power of lens, for refractive power of lens to increase by degree of 15 diopters, the shape of the lens changes from dash to dash. Increase by 15 diopter. It's again that power based question. So now there is an increase in 15 diopter. So the shape of the lens changes from what to what is the question. Option A, very convex to very concave. Option B, very concave to very convex. Option C, very convex to moderately convex. Option D, moderately convex to very convex. So if you're going to eye doctor, we can see they recommend some power, right? Sometimes it's in minus, sometimes it's in plus. Depending on that, they define which kind of lens they should use. So here they in the question they are asking increase in degree of 15 diopter. So what will be the correct answer? You are saying very convex to moderately convex. Let's see, okay? What is the correct answer? So it's an increase. It's correct when plus sign means convex, but it's an increase of 15 diopter. So the correct answer will be very convex to moderately convex means convex that is decreasing, right? So increase in 15 diopter. It must be convex. So that's why the correct answer is moderately convex to highly convex. Very convex. So diameter of the lens increasing means lens are becoming thicker and more curved. So this is because the higher diameter values means the lens can bend light more sharply. You didn't ask anything. So yeah. So increase in diameter means lens becoming more curved means higher diameter value always means lens can bend and lights more sharply, focusing it at a closer range. Okay. So you are right, it must be convex, but remember it's an increase in 15 diopter. So it must be increasing from the moderately convex to very convex. Next question. Night blindness is caused by loss of vitamin A, which leads to decrease in. This also we discussed in the introduction. If vitamin A is decreasing, some pigment in the something is getting lost. That's why our parents tell to eat as vitamin A containing food for increasing vision. So what is this pigment that is related with vitamin A? Option A retinal, option B vitamin D, option C melanin, option D pons. What is the correct answer? Okay. So when we said about rhodopsin, remember rhodopsin has two components. It is made up of opsin plus something else. So what is that something else we talked? And also that the clue is that word is very connected to the one of the layers where photoreceptor cells are present in the eye. So what will be the correct answer? Correct answer is retinal. Okay, retinal. So retinol and retinol esters are often referred to as preformed vitamin A. So the retinol, the name derived from the word itself, retinol. Okay. So retinol can be converted by the body to retinol, which can be turned to oxidize to retinoic acid, the form of vitamin A known to regulate gene transcription. Okay, so vitamin A is a source, retinol is a source of vitamin A. So if vitamin A is decreasing, then it will impair our night vision. Next question. This is we discussed in the last lecture, week 8. Pheromone are sensed by which sense of ours? Option A, auditory. Option B, olfactory. Option C, vision. Option D, haptic. Pheromones. We discussed about ants, how they maintain the root paths, how each ant follow the next ant in a single line, how they communicate. So that sense is caused by which sense in our body? Okay, you are saying different answers. Oh, sorry. This was C. Okay. I'm sorry, I didn't see the rest of the answers. I was stuck at that 6:31. Okay, so you are saying olfactory. Very good. Pheromones are caused by, sensed by, definitely by olfactory senses. Okay, very good. So how they are sensed to the brain? Because chemoreceptors are present in the olfactory organ, especially inside the nose. Then they are G protein coupled receptors and can be classified into two major categories: odorant receptors and pheromone receptors. So pheromone is sensed by pheromone receptor, and the smell molecules. Suppose this is smell of a spray or smell of very bad smell of something. Some wastes are there, so we detect a very bad smell. That is true odorant receptor. If it's pheromone, then it is true pheromonal receptor. Next question. Which of the following is not a cell type found in human eye? Uh, Options are option A bipolar cells, option B ganglion cells, option C amacrine cells, option D goblet cell. Which is not found in human eye is the question. Not a part of human eye. Yeah, this image, let it be there. Yeah, very good. So the correct answer is goblet cells only. Can you tell me goblet cells can be seen in which part of our body? What is the function of these cells in our body? If all others are found in eye, where we can see goblet cells, or what are the function of the goblet cells? Any idea? Okay. So you're right. Goblet cells are not found in eye, but all the other three bipolar, ganglion, and amacrine. You can see right here. Let me take again the laser. Here. So horizontal cell followed by amacrine cell followed by bipolar cell followed by ganglion cell. So both all the three Amacrite, bipolar, and ganglion are part of human eyes just before retina. These all layers helps in providing nutrition to the retinal pigment present, the eye present, and also they helps in to prevent the photo bleaching everything. Okay. Now, goblet cells. Goblet cells are not present in eye, but you can see them in intestinal mucosal epithelium. Okay, so they are the ones who secrete and synthesize and secrete mucus. So why mucus is needed in the intestinal epithelium? Because they are definitely cells and enzymes that are digesting the food. So in that digestion of process, if mucus is not present there, there is in the stomach also we can see strong HCl. So that can also degrade our cell lining. So in order to protect from that, mucus is there. Okay. So goblet cells are the one who secrete this mucus and that gives a protective role to protect our tissue layers from the acid produced by the stomach or to neutralize the activity of acid produced in the stomach. So photoreceptor cells, rod detects dash. So rods detect dash. Again, this can be asked by multiple ways. Sometimes question comes, what is the function of rods? Or this type of thing is detected by dash cell. So please remember to thoroughly understand the concept of what is the function of rods and cones. Okay. So here the question is about rod cell. Option A, intensity of light. Option B, color of light. Option C, both A and B. Option D, none of the above. So what is the function of rods? This we just discussed in one of the previous question also. So if you remember which type of cell is responsible for color of the light, is it rods or cones? Question is only about rods, not about cones. So what will be the correct answer? Hmm. Okay. Correct answer is intensity only because cones, there are multiple cones depending on which color. There are all the primary colors. Depending on the primary colors, we have three types of cones in our eyes. So, but rods, irrespective of that, it doesn't have any subtypes or it cannot detect any color, but it can detect, it responds to the intensity of the light. So, correct answer is A only. Activation of photopigments by light sends a signal via hyperpolarizing the rod cell, leading to the rod cells not sending its neurotransmitter, which leads to the bipolar cell then releasing its transmitter at bipolar ganglion synapse and exciting the synapse. Now that's just opposite question. I don't even have to read the question, I guess, but still I will read. Photoreceptor cell cones detect. Now it's the question is about cones. Again, options are saying. So what will be the option? Yeah, Austin is saying option B. Harini, you can also type your answer in the chat box. Yeah, photoreceptor cells cones detect dash. Cones detect color of light. Option B. Okay. So now, have you heard about color blind people? That's because they have not all the three consoles. Maybe some subtypes are not present. That's why maybe they if they don't have that particular type of console, maybe then they see most of the colors in some same color only. So they are known as color blind people. And without all the three, if they don't have any color, like any concepts, types, subtypes at all, then they cannot see color at all. Everything they will be seeing in black and white only. So they are completely color blind. During, again, the same question, repeat. During an eye transplant, what is transplanted or replaced? Option A, whole eye. Option B, retina. Option C, cornea. Option D, lens. What is transplanted or replaced? What is the correct answer? Eye transplant is the question. Okay, eye transplant. During eye transplantation, uh, read the questions carefully. That will be helpful for the exams also. Harini, do you have any answer? Correct answer is cornea. Okay. We multiple times said, and the class also sir said in the first lecture of the week 9. Cornea is the part that transplanted during most of the eye transplantation. So it's not about entirely taking an eyeball and keeping it somewhere. It's just about taking the, even though if like that person's eye transplantation is happened, even we after seeing we didn't even know that maybe his eye is transplanted or not, because this is just a part of cornea is transplanted. So it's also known as, this operation is also known as keratoplasty. Okay. Again, the question is about disorder, cataract. Cataract leads to dash, and during surgery, it is replaced by dash. Option A, blurred cornea, artificial cornea, replaced by artificial cornea. Option B, cataract leads to cloudiness of lens, and during surgery, it is replaced by intraocular lens. Option C, cataract leads to retina damage, and during surgery, it is replaced by intrarenal retina. Option D, cataract leads to pupil dilation, and during surgery, it is replaced by Botox. What is the correct answer? Cataract leads to what, and during surgery, it is replaced by what? Is the question. So Austin is saying option B is the correct answer. 6:53 and 6:54 till there is time. So then we'll discuss. 6:54. So correct answer is yes, good. Cataract leads to cloudiness of lens. That's why when our grandparents and all, we can like they cannot see properly, right? So because they're, even we look into their eyes, we can see the white part. That color is like, that color itself is faded. So if the cataract is there, then they in the surgery it is replaced by intraocular lens. That is correct. So this intraocular lens is an artificial lens. It's also we generally name it as IOL also. Okay. So first, what do doctors do in this surgery? Remove that lens, whatever the cloudy lens, and replace it with this artificial lens. This is a very common kind of surgery. Also, we can see most of the elders do this surgery. At the blind spot, there are dash photoreceptors. Option A maximum, option B no, option C half the density of, option D horizontally aligned. What is the correct answer? At blind spot, there are dash photoreceptors. The name itself says it's blind spot, which means there can be how much photoreceptors? I think this also we discussed in one another question earlier. So there can be how many photoreceptors? What is the correct answer? Blind spot is the earlier question was what is the area called as where most of the nerves leaves and goes to the optic nerves leaves from the area. That is blind spot definitely. So when we discussed that answer, we told about this also. How many photoreceptors are there? Correct answer is no photoreceptor in blind. That's why the name comes blind spot because there are no photoreceptors. So correct answer is option B. Okay. So the natural blind spot in the visual field has been known as a large oval region that cannot receive any optical input because it corresponds to retinal optic disc containing no rod or cone photoreceptors. Next question. This this answer is a part of the one of the earlier question we discussed. Dark current is dash. Option A depolarizing. Option B hyperpolarizing. Option C either A or B based on the color of the light. Option D either A or B based on the intensity of the light. Dark current is dash. Is the question. Any idea? Dark current is what? What happens when dark current comes? That's why sodium and calcium are coming. That's why it is maintained at minus 40. So it must be what? Yeah, correct answer is depolarizing only. Why? As we discussed, in the dark, CGMB levels are high and keep CGMB gated sodium channels open, allowing a steady inward current called dark current. So in the dark current, CGMB always open. That will allow more sodium to come inside. So this dark current keeps the cells always around minus 30 millivolt, leading to glutamate release. Lights leads to reduced CGMB levels and closure of CGMB gated. So whenever light is there, the opposite process will happen. Whenever light is there, it will make sure that CGMP is closed and no more sodium is coming inside. So dark current is always depolarized. Next question is resting membrane potential of photoreceptor is option A minus 90 millivolt, option B minus 40 millivolt, option C minus 75 millivolt, option D plus 90 millivolt. What is the correct answer? We'll wait till 7-1 or 7-2. Uh, okay, Austin said. Austin is saying option B is the correct answer. Is there any other answer? We discussed just now only, right? Darker end means CGMP is open, which allows sodium to come inside. That maintains a resting membrane potential of dash. Correct answer is minus 40 millivolt only. Very good. So I don't have to explain, right? If you want any doubt, remember this class lecture where sir draw this. Where is my this thing is going? Sorry. Uh, Here, if you're checking when the light happens, comes, if you're checking the voltage, it is minus 40 millivolt. So photoreceptor resting membrane potential is minus 40 millivolt. Which of this is not a part of the eye? Option 1 iris, option 2 cornea, option 3 hair cells, option 4 ciliary muscles. Which is not a part of the eye? I'm really sorry about my cold. Which of these is not a part of eye? Symbol only. Iris, cornea, hair cells, ciliary muscles. What is not a part of the eye? Hair cells, right? Hair cells are not a part of the eye. Hair cells, we talked about hair cells in the ear. Whenever there is pressure or sound or vibration or something comes, hair cells start detecting and it starts moving. There are two types of hair cells, gynoecilia, stereocilia. So this vibration makes the vibration of the hair cells. As a result, the movement happens. As a result, the vibration detected, then it will leads to the auditory like center of the brain. The information will be passed. There is organ of Corti. Inside that there are two organs which is connected to the auditory center. Remember all these things. Last week we discussed. So hair cells are not a part of eye. Okay. And if you want to see where is iris, iris are this muscle like part of this part is iris. Cornea, cornea is that our white part. Ciliary muscles here. Lens is connected to the layer by through the ciliary muscle. The liquid filled between cornea and lens is called as dash, whereas from lens to retina is called as dash. So liquid filled between cornea and lens is dash and lens to retina is dash. In the week 9, first lectures are draw the structure of the eye in that it is there. What is the part known as between cornea and lens? Next question is what is the part that is between lens and retina? Option 1 aqueous humor. Both are aqueous humor. Option B one is aqueous humor, second one is vitreous humor. Third option is both are vitreous humor. Fourth option is first one is vitreous humor and second one is aqueous humor. So what comes where? Any idea? So correct answer is option 2. Okay. Liquid filled between cornea and lens is called aqueous humor, and lens to retina is called as vitreous humor. So correct answer is option 2. Okay. Uh, this here you can see, right? This is cornea, and between cornea and lens, whatever is there, that is aqueous humor, and from lens to retina, there is vitreous humor. During eye donation, what is removed from the eye for transplantation? Same concept, third question. During eye donation, what is removed from the eye for transplantation? Option 1 whole eye, option B retina, option C lens, option D cornea. What is what? Correct answer is cornea, right? So during eye donation, what is removed from the eye of the transplantation? Correct answer is cornea. Which of this is the best description of cataract? Option 1 lens becomes shrunk. Option B lens becomes swollen. Option 3 lens become clear. Option D lens become blurred or cloudy. What is happening during cataract is the question. Yeah, so the correct answer is lens become blurred or cloudy. Very good. We remember what is happening. Misfolding of protein alpha crystalline is happening. That's why lens is becoming blurred or cloudy because the protein which was already transparent and everything due to the misfolding of protein it will appear as a blind spot. So that's why lens is becoming blurred or cloudy in cataract. Which of these enzyme is linked with formation of island? This also are talked. So first there was lens where like normal cells only which contain all the organelles, but during the course of time all the organelles degrade due to the a high activity of one enzyme. So what is that enzyme? Option 1 lactase, option 2 15 lipoxygenase, option 3 alcohol dehydrogenase, option 4 pepsin. So what will be the correct answer? What is the enzyme? Any idea? Correct answer is if organelle is there, membranes are all made up of phospholipids. So in order to digest that, what should be there? After digestion, alpha crystalline comes and fills up the entire lens. So before that, all the organelles should be removed. For that, correct answer is lipoxygenase. Okay. Remember the first class. Sari was talking about lipoxygenase. So due to the high activity of lipoxygenase, the lipid membranes all get dissolved, and that's how structure is. The organelles are removing, and alpha crystalline is filled up. That's why lens is transparent. Next question. Features of retinal epithelial layer. A. So this is about features of retinal epithelial layer. Which layer is this? According to the peculiarities they are saying, you have to tell. Okay. First point is they can provide nutrition to rods and corn cell. Option B, they prevent rods and corn cells from photo damage. Which of the following are correct? So the question is about retinal epithelial layer. What are their correct function or functions? So is it only A is correct? Only B is correct? Both A and B are correct? For none of the above are correct. None is function of A. So what is the correct answer? What are the correct options? Yeah. Austin is saying both the answer are correct. Very good. Yes, both the answer are correct. Retinal epithelial layer also, so I discussed in the first class itself. They provided nutrition to photoreceptor cells as well as they helps rods and ponds from photo bleaching. Otherwise, what happen? Due to the light, whenever it comes, this due to the disintegration, reactive oxygen species can get accumulated inside the eye, which leads to bleaching, and lot of photoreceptor cells get distracted in that way. So in order to protect from that, there will be should be a protective layer that stops this much high intensity of light rays to come into the retina. So that layer is the retinal epithelial layer. So both A and B are correct. Okay, next question. Rods and cones cells are only two options. Either they electrically active or are they electrically inactive. What is the correct option? One or two? Question is saying one. Yes. So or no. One option is electrically inactive. If it's electrically inactive, then think about the CGMB things we told. Uh, both are active only, right? That's why they are able to detect That's why they are called as photoreceptors. So whenever the light rods can detect the intensity of light and cones can respond to the color of the light. So whenever light comes, rod cells disintegrate into retinal and opsin. That will make the some enzyme get activated. That will go and tell the CGMB to get it closed. So both are electrically active. Okay. The absorbance of rods. You remember what are electrically active cells which can generate action potential? Otherwise, all excitable cell, non-excitable cell. Please go back and remember the old lectures also. Okay, don't forget. The absorbance of rod speaks at wavelength of around. Remember, rod is what kind of photoreceptor cell? It can detect what? So the wavelength peak around what nanometer? Option 1 200 nanometer, option 2 300 nanometer, option 3 400 nanometer, option 4 500 nanometer. So what is the correct answer? Austin is saying option 4 500 nanometer. Yes, that is the correct answer. It's always found that Rhodes activity peaks at the around 500 nanometer. Okay, so there is a paper also that they have actually experimentally proving that it's the rod's activity peaks at 497.6 plus or minus 3.3 nanometer, which means that is closer to 500 nanometer. So correct answer is 500 only. Which of these light mostly lie in the wavelength region of 400 to 700? I think we have learned this in 8th or 9th, or, depending on the syllabus, obviously. But we have definitely studied it in the school itself. 400 to 700 nanometer. I have given the answer also in the figure. So you can look it and say, what are the rays that comes in the range of 400 to 700 nanometer? Option 1 ultraviolet, option 2 visible, option 3 far red, option 4 none of the above. 400 to 700 nanometer. The name is written here. What is this? 400 to 700 and the name is here. So is it far red? What is written on here? This rays comes in the range of 400 to 700, 400 close to blue and 700 comes in the range of red. Before this ultraviolet, after this infrared. So 400 to 700 is visible, right? Visible light comes in the range of 400 to 700 nanometer. So 2 is the correct answer. Okay. Visible light comes in the range of 400 to 700. Again, same question. Resting membrane potential of photoreceptor is around dash. I know now you guys know because we already discussed. Is it minus 80, minus plus 80, minus 40, plus 40? Yeah, correct answer is minus 40 only, right? I don't have to again go and discuss. Correct answer is whenever minus 40 millivolt. Option 3. So, why this is happening? The presence of cyclic ganosin monophosphate. So, CGMB's full form is that cyclic ganosin monophosphate. So, they always keep cation channels open in eye. That's why the influx of sodium or calcium occurs. Then this depolarizes the cell to about minus 40 millivolt, and that current is known as dark current. After a result of this, glutamate releases because it is at minus 40 millivolt. Then as a glutamate release, as a result, neurotransmitter glutamate releases. When light strikes the photoreceptor, it triggers a cascade that reduces the level of CGMB. That's what we said. It will become retinal opsin, then retinal that will activate some of the enzyme that will go and tell the CGMB to keep it closed, which will lead to the closing of sodium and cation channel, calcium cation channel get closed. This stops the influx of positive ion, which result in the photoreceptor becoming hyperpolarized or more negative. Okay. Uh, the hyperpolarization caused by the light reduces the release of glutamate, sending a signal to downstream neurons. Like that, information get reaches in the optic center of the brain. Yeah, so that was the last question, and I hope you are doing all days assignment questions. And also, please be sure that to go back and like the last question last week, I think last week only there was a revised announcement of some assignment solution were not correct. So I discussed that in that live session itself. I think it was a question from second week and week 5, I guess. So like that, if you have did the last week assignment solution also, you can see there are two wrong answers they have published. I think they will later tell the correct answer. Should I go and open it? Let me open it. Okay. Yeah, this one. Yes, Ritu, you can ask. Actually, I don't know about the technical details of exam, if it's regarding subjective or objective. So it's better to ask to the NBTL team only. Whoever contacting through the mail, you can ask. First, you can ask the question. If I know, I will definitely tell the answer. I cannot hear you, Ritu. Yeah, here. This is correct. Okay, they have already edited it, I guess. This is fine. Hair cells. I saw this as different one. This is correct one. Yes. Again, let me go through one by one. What is the sensory receptor muscle that detects changes in length? Muscle spindle. Yes. Intrafusal fibers, muscle spindle. What neuron carries signal from muscle spindle to spinal cord? Uh, yeah. Sensory neuron. Polysynaptic, correct. What motor neuron innervate extrafusal fibers? Alpha. Mostly which neurotransmitter is released at noracetylcholine, correct. Calcium, correct. Okay, but I think in the explanation it is correct, but if you are going and looking at the released question, where is it? Here. Here. So in the released question, they have given the correct answer, right? The process linking electrical excitation and muscle contraction is called a excitation contraction coupling is the correct answer. They have given in the explanation as correct answer as excitation contraction coupling only. But in the answer, they have given wrong answer. So please go to the solution and check, right? That is correct, but in the when the answer is released, that it got changed. And here, what are the sensory cells in the cochlea called? Correct answer is hair cells only, right? In the explanation also they are saying hair cells are the sensory cells that present in cochlea. We have seen in the last week, but when the answer released, they counted it as wrong. So that part got interchanged. So I think they will edit it and release the again with new answer. Okay. Yeah. So is there any other doubts to ask? I wanted to tell this because I noticed this. Is there any doubts to ask? Ritu, you wanted to ask something? You can ask. Are you there? Hello. I'm audible only, right? Yeah. Are you typing your query? Oh, okay. You already written. I think it's both objective and subjective. It's not like exactly like assignment part. There will be both objective and subjective type. For more details, like how much part is objective and subjective, today Austin also asked me. That I don't know. So I think you have to better will be asking NPTEL team. Send a mail to them. They will reply. Okay. Yeah. So it's already week 9 now. Now we have only week 10, week 11, and week 12. Then there will be a final session where we can discuss all the some of the questions from mostly covering from most of this. So that's it. Yeah. So see you in the Friday with week 10 live session from 6 to 8. Okay. Bye. Take care. See you. No doubts, right? Can I leave the meeting? Bye bye. Bye, Austin. Bye, Ritu. I think others left. Yeah. Bye bye.